So, welcome to the new episode of the Microbiology Tube. So, today the topic is the contamination, preservations, and the spoilage of milk and milk products. So, what are the sources of the contamination? So, one of the sources of the contamination is on the farm. So, the next source of the contaminations is in the transit and at the manufacturing level. So, if we see what are the reasons for the contamination of the microorganisms in the farm. So milk contains relatively few bacteria, so when it leaves the odor of the healthy cow, and generally this bacteria do not grow in the milk. So during milking, the contaminations of the milk is by the exterior of the udder and the adjacent areas. The bacteria found in the manure, soil, and water may also enter from this source. So two most significant sources of the contaminations are the dairy utensils and the milk contact surfaces, including the milk pile and milking machines. Undesirable bacteria from the sources include the lactic streptococci, coliform bacteria, psychotropic gram-negative rods, and thermodurics, which survive the pasteurizations. The example of these bacteria are Micrococcus, Enterococci, Bacilli, and the Brevibacteria. So other possible sources of the contamination are the hands and the arms of the milker or the dairy workers, the ear of the brown or the milking parlor, and the flies. The number of the bacteria per liter of the milk added from the various sources depends on the care taken to avoid contamination. So in transit and at manufacturing level. So after the milk is left in the farm, the possible contamination includes the tank trough, transfer pipes, sampling utensils, and the equipment at the market milk plant and other processing plant. So pipelines, vats, tanks, pumps, valves, separators are the possible sources of bacteria. The amount or the level of contamination from each of these sources depends on cleaning and sanitizing methods. Hands and arms of the employees are a possible source of contaminations and pathogens. So there are different techniques for the preservations. One is the asepsis. So asepsis means not letting the microorganisms enter inside the milk. So next is the removal of the microorganisms. So there are various methods of removal of the microorganisms. So one of the best techniques is the filtration. So in some products the centrifugations is also used for the removal of the microorganisms. Next is the use of the heat. So by the use of the high temperature you can remove the microorganisms. So in the milk, pasteurizations are major used for the removal or to inhibit the bacteria. So use of low temperatures around 4 degrees centigrade are also used. So next is the drying, and next is the use of the other preservatives. So let's come to discuss about the spoilage. So the milk is an excellent culture medium. As we know, the milk is an excellent culture medium because it consists of the moistures, the pH is near the neutrality, and these are rich in the nutrients. So it is the food for the many kinds of the bacteria. So whenever we store the milk, the bacteria like the Pseudomonas, Acinetobacter, Aerobacter, Alkaligens, and the Flavobacterium, as well as some members of the Enterobacteriaceae are found. So when we extend the storage temperature for a longer period of time, then psychophilic or psychotropic organisms causes the major spoilage. So next, the spoilage of the milks usually is caused by the psychotrophs that recontaminates the milk after the pasteurization. Also involved are the thermodynamic psychotrophs as well as heat stable protease produced before the pasteurization. So after the pasteurization has been done, a lot of the psychotrophs will be inhibited, but the major contaminants by the psychotrophs are those types of psychotrophs which recontaminate the milk after the pasteurization. And not only this, there are certain protease enzymes so that are produced before the pasteurization, and that are the heat stable proteases. So these heat stable proteases are responsible for the spoilage of the milk. So next is the gas formations. So the chief gas former are the Clostridium bacteria. So and Clostridium bacteria, that is followed by the Bacillus. So the chief gas formers are the coliforms, Clostridium, and the gas forming Bacillus that will both the hydrogen and the carbon dioxide, yeast and heterofermentative lactase that produce only the carbon dioxide. So gas production evident by foam at the top of the milk and is supersaturated with gas by gas bubbles in the curd, by floating curd containing the gas bubbles, or by ripening a part of the curd by rapid gas production, so causing the so-called stormy fermentation of the milk. So you can see in the figure some of the stormy fermentations. So now the major bacteria is the coliform. So the coliform bacteria are responsible for the gas formation. Heterofermentative lactase also may produce the gas, but usually not enough to be evident. So yeast usually are absent or in low numbers in the milk and do not compete well with the bacteria. Bacillus and Clostridium do not compete well with acid formers at high temperature, but may function if the acid formers are absent. Thus, in pasteurizing temperature or above, the spores of the Clostridium and Bacillus will survive, and gas formations by the spores may take place. So next is the proteolysis. The hydrolysis of the milk proteins by microorganisms is usually accompanied by the productions of bitter flavor caused by some of the peptides released. Proteolysis is favored by the storage at the low temperature, by destruction of lactic and other acid formers by heat, and by the destructions of formed acids in the milk by molds and film yeast, and neutralizations of the acids by products of microorganisms. So actively proteolytic bacteria are found among the species of Micrococcus, Alkaligens, Pseudomonas, Proteus, Flavobacterium and Serratia, so all of which are genera of non-spore forming bacteria and of genera Bacillus and Clostridium of spores former. Some species of genera Micrococcus, Pseudomonas, Proteus, Flavobacterium, Alkaligens and Bacillus can grow at low temperatures and hence are likely to cause some proteolysis and or bitterness of the milk held in chilling temperatures. So next is the ropiness. Ropiness or sliminess can occur in the milk, cream. So bacterial ropiness is caused by slimy capsular materials from the cells and ordinarily develops best at low temperature. The ropiness usually decreases as the acidity of the milk or the cream increases. So two types of the ropiness are there, that are the surface ropiness and ropiness throughout the milk. So surface ropiness is caused by the alkaligens vicolactase and organisms chiefly from water or soil that can grow fairly well in the vicinity of 10 degrees centigrade. Ropiness throughout is caused by Enterobacter erosions, Capsella oxytoca and really the Escherichia coli. Sources of the bacteria causing ropiness are the water, manure, utensils, and feed. The reductions or eliminations of contamination from the source helps prevent ropiness. So next is the changes in the milk fat. So milk fat may be decomposed by various bacteria, yeast, and the mold. So oxidations of the unsaturated fatty acid results. So that yields the aldehydes, ketones, and acids, and results in the tallow odors and taste. So next is the hydrolysis of butter fat to the fatty acids and glycerol by the enzyme lipase. So the third or the last is the combined oxidation and the hydrolysis to produce the rancidity. So species of lipase forming bacteria are often found in many of the bacterial genera. For example, Pseudomonas, Proteus, Alkali, Bacillus, Micrococcus, Clostridium, and many other. Pseudomonas fragi and Streptococcus aureus produce fairly heat resistant lipase which may survive pasteurization if present in the raw milk. So alkali production is also there in the spoilage of the milk. So the microorganisms responsible are the Pseudomonas fluorescens and the Alkaligens picolactis. So this results in the productions of ammonia from the organic acids, and urea results in the alkali formation. So the next is the flavor changes. So sour or acid flavor, so that is produced. So next is the clean, so that is produced by the Streptococcus lactis or other lactics. Aroma is produced by the, or aromatic odor is produced by the Streptococci and aroma forming leuconostoc species. Sharp flavor is formed by the coliform bacteria and the Clostridium species. So clean and the aromatic flavors are desired, but fermented milk products but soft flavors are undesirable. So bitter flavors result from the proteolysis, lipolysis, or even the fermentations of lactose. So organisms mainly used for the bitter flavors are the coliform bacteria, yeast, and the actinomycetes. So bond flavors, that is mainly produced by the Streptococcus lactis variety, that is the multigens. This produce the bond flavor. And the changes of the milk color, so the milk will be changed into the blue color by the Streptococcus sansinia when grow with Streptococcus lactis. So yellow milk will be produced by the Pseudomonas syngenta and the species of Flavobacterium. So the red milk will be produced by the Serratia mercensis and the Micrococcus roseus. So brown milk will be produced by the Pseudomonas putrefaciens.